So I'm going to be talking to you about, some of the stuff that we've done using, transcriptomics, both bulk, single cell and spatial sequencing. Not a small topic to talk about in 17 minutes, but I'm going to show you some of the stuff that we've done and how these technologies kind of work and what are the strengths and weaknesses and how you can, in a way, leverage those. So I really like this image of the different types of transcriptomics. You're kind of really looking at an organ here. It's shown like a brain, but it's in like Lego bricks. And if you think about how bulk RNA-seq kind of works, it's incredibly sensitive. You kind of see all the pieces. So it's a very high resolution, high sensitivity, but you have absolutely no idea where each one is kind of coming from in the tissue. More recently, we've had the single cell sequencing that has really transformed the way that we can interrogate disease processes. So you can see, the gene expression in individual cell types in the disease process itself, but it's a moderate resolution. So instead of getting everything, you get maybe 2,000 to 3,000 genes per cell. You don't capture everything that the cell actually expresses. And then very recently, we have had these spatial RNA-seq technologies that can tell you exactly where in the tissue the gene is being expressed. Incredibly powerful technology, still in its infancy, and I'm going to show you some more data on that. But the resolution is still very limited and usually in the range of 200 to 300, 400 genes per cell. So you have to kind of be creative how you use these technologies to really kind of gain insight into disease mechanisms. So I'm going to focus a little bit on the spatial RNA-seq. So this is really what you're trying to do, is to kind of take a tissue like you have in psoriasis, and then you run it on what we call like a spatial profiling kind of a platform. What I have here is what we call the Xenium platform. This is a fairly high resolution platform. It's in situ technology. And it can interrogate about 5,000 genes, and here we have an added 100 gene panel on top of it that is more customized to skin. But you can use this as a way, just transcriptomically, to kind of recreate the entire disease and the tissue structure, like you can see here, with the different kind of cell types based on just the differences in the gene expression, kind of shown in different colors, and you can really see how the structure is kind of preserved, even if you're just looking at gene expression itself. So if you think, nowadays there are at least 10 different spatial profiling technologies. They're all a little bit different. Some are sequence-based, some are probe-based. And this is kind of the one that most of the field is currently using, which is the in situ platforms. There are a few of them available. One of them is Cosmex, another one is Murphys, the other one is Xenium, and this is the Xenium platform. But what these technologies are based on is that you have a tissue section. It can be formalin fixed or it can be frozen. You make a section, you cut it onto a slide, and then you do this fixation and permeabilization, and you kind of amplify with this probe hybridization, ligation, and amplification to kind of make the target a little bit bigger. And then what the machine does, like the Xenium Analyses, it kind of run continuously these cycles with the different fluorescent probes to kind of detect the genes in the tissue. And then you have the machine in the end kind of take these different cycles at different colors and then reconstruct that into what's typically is a massive data file. And then you can use that data to kind of reconstruct the disease processes that are going on in the skin. If you look at, just looking at specific genes that are kind of localized, limited to specific cell types, you can really appreciate what kind of a view and resolution it can actually give you on the tissue. Here we have T cells, CD4, CD8 cells. Here we have the Tregs. You can see where they're localized in the disease process. You can even detect B cells that are fairly rare in psoriasis, macrophages, and even dendritic cells. And same thing here. Here you have HLA-C expression in the skin. You can see it's very heavily localized to the epidermis. IL-23 also, you find most of it being expressed in the upper layers of the epi. If you're thinking about where that actually is predominantly coming from. IL-17A and F, interferon gamma, IL-36, and then CCL20, the chemoattractant 4 Th17 cells. So it can give you a very broad view and fairly sensitive view of what's actually happening in the tissue itself. But you don't really want to have this technology as a way to just generate pretty looking pictures. What you really want to do with these technologies is to really use them as a way to get biological insight into what's happening in the tissue. And it's a fairly complex way to kind of analyze, and we put a lot of effort into kind of optimize like a processing pipeline. But you need to do cell segmentation to kind of see where the boundaries are between the cells, and you need to know the X and Y location of each one of those gene transcripts, and then you need to kind of localize that to the cell type itself or the cell boundary. And then you need to decide if you're, the transcript profiles, you need to do normalization and quality control over the data. Then it's a question if you have single sample or multiple samples, how you kind of integrate them. You do principal component analysis with batch correction, dimensional reduction, and then you do clustering to try to define the cell types. And then when you have kind of done this deconvolution of the data into like cell types, then you go back to the image and you reconstruct the disease process using what we call in situ plot typing. And then you can do the downstream analysis, which you call like domain detection, cell networks, and intercellular interaction. So what you really want to get to is what I have here, is really how these cells are interacting in space. And you want to see the ones that are diffusion dependent, the secreted chemokines, the secreted cytokines, and how they're interacting over a distance, or the ones that are contact dependent, where you have a direct interaction between something on the surface of the cell against the surface of the adjacent cell. And then what you can do is you can look at the signaling strength and the communication across the distance in the tissue using this technology. And you can see using RNA velocity and spatial positions, you can look at the interactions between the different cell types. You can see how the expression changes and the slope of the changes, and then you can reconstruct the tissue where you're looking at a very specific signaling, and you can see where the signaling is actually increasing and where it's actually kind of being shut down. And this is kind of one way that we have done this for psoriasis, looking at just type 1 interferon and type 1 interferon signaling. We have the center on the bottom, and then you have the receiver on top, and you can kind of make a connection between the different cell types, how they're interacting, and which cells are predicted to be communicating in the disease process. So incredibly powerful, and it's really going to transform the way that we look at psoriasis and any other disease that you actually want to focus on. Another thing is the single cell, which also is incredibly powerful technology to kind of look at biology, and you can get even more out of it if you integrate that with the spatial sequencing. So you use the signatures that you have from the single cell, you integrate that with the spatial, and then you can kind of reconstruct the tissue like I have over here, where you have the different subtypes of keratinocytes on the epidermis, and then the various cell types on the bottom. And this is also a very effective way to kind of look at cell-cell communication, baseline post-treatment, and then you can kind of compare that to non-lesional baseline skin. And what I have there in the black box is just the myeloid and T-cell interactions with epithelial cells. You can see it's very highly enriched, shown in red, 150 plus interactions that are predicted. And then as you kind of treat the disease, you can really see that interaction, that communication network that is disease-associated really collapses and goes away. Another thing is how you can integrate these data with almost any type of data set that you actually have. Here what we have is to integrate the genetics, the data from the GWAS, the predicted susceptibility genes that are involved in psoriasis. And here you're looking at the different cell types in lesional and non-lesional skin. So you can see the red spots, that's where there's gene expression detected for those particular risk genes that I have on the other y-axis. And then you can see that there's a cluster of the susceptibility genes mapping to T cells. There's also quite a few mapping to dendritic cells, keratinocytes, fibroblasts, and interestingly, we have a cluster here that maps onto melanocytes. A cell population that has been studied, but is still massively understudied in the pathogenesis of psoriasis. You can also look at it in terms of the different signaling. So here we're using bulk RNA sequencing of cytokine-stimulated keratinocytes. And we're looking at the downstream signaling, the genes that are induced in keratinocytes by these various cytokines, and then linking that back to the different genetics. So we're looking at the log FDR of the enrichment and then the observed to expected ratio. You can really see that many of the genetic risk factors for psoriasis actually align with the same genes that are downstream of IL-17A and TNF. You also see quite a few genes that align with interferon responses and then IL-36. So in a way, you can kind of define these three major kind of cytokine or four cytokine axes as the mainstay, the critical key cytokine pathways in psoriasis. And you can also do this by looking at the different kind of clinical features of psoriasis. So you can use these types of data, these types of data sets that you have to really kind of get a better understanding of what really is happening in psoriatic skin. So here we have the different components of the PASI, the scaling, the thickness, the redness, and we're looking at different severities. So you have an absent, mild, moderate, severe, and very severe. And then we use the same kind of a cytokine response signatures to kind of really compare what is being enriched. So what you see here we're comparing disease versus normal skin, and then on this axis we're comparing severe lesional types versus moderate lesional types versus mild lesional types. And the most severe ones are there far on the left. And when you do it this way, if you look at IL-17A signal, you can really see that it's really defining the presence of the disease, more so than the different severity of the lesions themselves. Same with the interferon, type 1 and type 2 interferon. It really is kind of defining the disease process, less so with the severity. And then IL-36 is really what is going with the more severe lesional types. That's probably where it's being activated, that's where it's most active. So in a way, you can kind of reconstruct and have these cytokines and these transcriptomic signatures as a way to help you get a better understanding of what really is driving the disease and driving this different phenotypic variation of psoriasis. So IL-17A and TNF have to do with disease, to some extent severity. Interferons really are associated with the disease itself, not so much the lesional severity. And then IL-36 is clearly, as you get more severe psoriatic lesion, the more activity of the IL-36 axis you seem to have, based on these transcriptomic signatures. And then also, on the last couple of slides, I just want to show you how you can also use single-cell and spatial-seq to really get an idea of what might be driving different subtypes and types of psoriasis. So this is single-cell data that we generated from about 13 patients with generalized pustular psoriasis. So you can really see the different cell types that we can identify in the tissue. These are from lesional skin. You can see the different cell proportions that are kind of shifting as a part of the disease process. You can see that there's a massive increase in the myeloid cells. And then you can kind of look at individual subtypes of these myeloid cells to really kind of define, which ones are the ones that are going up and which ones are kind of going down, and where's the shift. And here you see that there's like 3 different types that are going up, and there's the one in red, that is really kind of getting less pronounced and almost disappearing. You can look at the different types in terms of their numbers, the proportion using just single kind of like t-test. You can look at the differentiation, whether it's more M1 or M2 macrophages, and then you can look at them in terms of like pseudotime trajectories, how these cells are kind of shifting as part of the disease process. And here you see that during the disease process, we're shifting towards a more like an M1-like path for the myeloid cells, towards the ones that are shown here, the IL-1 beta positive macrophages. You can also look at neutrophils. That's one way that these technologies can really help you to kind of look at, some of the key populations that can be hard to study otherwise. You can see we detect multiple different clusters of neutrophils, 3 major clusters, which of only one is found in GPP. You can look at and see that the subset, the CXCL8 or IL-8 positive neutrophil population, is highly pro-inflammatory. They have increased NF-kappa B responses, an inflammatory response. You can look at the different kind of clusters and trajectories. You can define the different branches, how they are actually operating in the disease. And then you can look at what are the molecular processes that these cells are really engaging in the disease process. And here you have response to molecular bacterial antigen, response to polysaccharides, neutrophil chemotaxis, and regulation of acute inflammatory process. The same thing with the keratinocytes. You can look at how these are behaving during the disease process. You can look at where that inflammatory signal is coming from. And there on the left, you can see the IL-36 score is really most pronounced in the spinous and the supraspinous keratinocytes in GPP. You can also see the same signal in IL-1 beta. And if you compare the IL-36 positive keratinocytes in either the spinous or the supraspinous layer, you can see they have a much higher inflammatory load than the ones that are IL-36 negative. And if you look at the incoming and outgoing cell-cell communication that are happening in GPP skin, you can see in normal skin that it's the IL-36 negative keratinocytes that are really kind of dominating the cell-cell communication. But in GPP, you're really getting a shift towards these IL-36 positive interacting with the neutrophils. And you get a network of a neutrophil keratinocyte crosstalk that is really shifted towards the disease state in GPP, particularly between IL-36 positive keratinocytes and neutrophils. Not surprising, this is kind of what you would expect given the genetics that we know about GPP, that it's very IL-36 centric, but it's really nice to see that coming out of a data set that is approached in a very different way. But really kind of helps you to kind of define what is really the core mechanism that is operating in a disease. So just to kind of briefly summarize, so I would say based on my experience with these technologies and hopefully what I've shown you with some of the data that I've shown you here is that these technologies are really going to transform the view that we have of biological processes in psoriatic disease. The limitations, though, with these technologies, they're extremely expensive, and you need a lot of infrastructure to be able to not necessarily generate the data, but to actually analyze it and make sense out of it. And then we are going to have all the types of single-cell data and multiple, multi-omic data that we need to further kind of integrate with this. And that includes chromatin data such as ATAC-seq, chromosomal looping data that we get from Hi-C, deeper dive into the genetics and how that kind of links to the biology, and then the microbiome, the metabolic changes, and a few other technologies that we need to kind of integrate. But I think this is going to fill in the gaps and really accelerate the path towards a cure in psoriasis, and I really believe that's possible. So with that, I'm going to end and if there's time, I'm happy to take questions.
We have time for one or two questions. Yes, we do. Yes, Paula, immediately.
You mapped the signal you got in IL-20. Very nice pictures. Obviously, they're not only pretty picture, I know that. Did you map the signal you got in IL-23 in the epidermis? Is it really keratinocytes? Is it Langerhans cells? What is it up there?
Based on the data that we have, it looks like it's keratinocytes.
Is it just IL-23p19, or do you have also?
It's the IL-23A, which is the P19.
But not P40?
No, that actually is quite hard to kind of detect that. But if we and others have actually run ELISAs on keratinocytes, and they make IL-23, but at very low level. And they make less per cell than dendritic cells or macrophages, but there's just 100 times more keratinocytes. So in the end, you're getting more IL-23, but it's probably also important, where it's being made and where it's being secreted. These are the same locations where you would expect to see the TRM cells in psoriasis lodged within the epidermis. And it's probably this low-level expression and secretion of IL-23 that is like a survival factor for those TRMs. And I think that's one of the reasons why the anti-IL-23 treatments work so well.
Any other question? Yes. Luis, thank you.
Amazing lecture. In that correlation or association between the severity and the expression of IL-36, was any difference when the severity was graded according to desquamation, infiltration, or erythema?
Oh, you mean break it down into like the different kind of components? Yeah. That's a great question. We actually haven't done that yet, but we clearly need to. My guess is that it's probably more the acanthosis and the scaling than the redness. Really? But I don't know. I haven't done it.
Thank you.
I clearly need to do it.
Back behind the pillar.
Yeah.
Oh, sorry.
you project as you look at more psoriasiform conditions or lichenoid conditions that you can't say that's psoriasis, but how close does it have to get, or how far can it get to still be calling it psoriasiform?
Oh man, yeah, that's not an easy question. Where do you draw a boundary from one disease to the next? That's the thing, is like, you know, these technologies are very good to kind of get an idea what's you know, driving a disease process in patients. To kind of address heterogeneity, you need a, you know, a much larger scale kind of an approach, usually than hundreds of patients. What we have done both on bulk RNA-seq and single-cell-seq, it's much easier to kind of think about these disease processes not as defined entities, but actually as a spectrum. And if you think about where we have the definition of disease, this is like a 19th century, early 20th century construct, what we have in the textbooks. And it's all based on clinical features, and that's how we define disease. But if you look at the biology behind it, it's much more complex and much more overlapping. And when you're in the clinic, you see these patients that are kind of these in-betweeners that you really have difficulty telling, you know, which way to go. And I think it's going to come down to the same thing when we start to kind of do this transcriptomics on a scale. And we're really going to see the same thing and see the plasticity of the response across different phenotypes. But we're not quite there yet. And again, it's usually the cost that is the issue.
Yes, one last question. JT, there. Wait for the microphone, JT, please. It's coming.
Well, you know, one of the biggest mysteries about the psoriasis is the cause of the epidermal hyperplasia. Have your studies yielded any insight into this?
Oh, that's not so much. We haven't really looked at that in terms of, but I think there are plenty of reasons. Obviously, the EGF, you know, ligands could be playing a role, and we know from the amphiregular mouse that turning that on can really give you a psoriasis-like phenotype. But I think for me, I like the involvement of the IL-20 family cytokines. And if you think about biomarkers for psoriasis, hyperplasia, IL-19 actually is a very sensitive marker for that, and in particular kind of a treatment response. So I think IL-19, IL-20, IL-22, maybe IL-24 are all going to have a role in kind of contributing to that, you know, that proliferative phenotype. But there's like always there's always something more, and I don't think we have a good grasp of what that is.
Thanks.